Hi, this is Miss Litton, and these are my wonderful AP biologists here at lunch for your last possible review for your Unit 2 exam. What I'd like to do is just make sure there aren't any big ticket items that you still want me to go over of these 4 chapters that we're going to discuss. I want to remind you that yesterday, thanks to some very faithful students, we went through all of the essay questions, and that is already posted on YouTube. So you can go through that and review your essays and some additional bonus material. Before I forget, I want to tell you, do you remember in the cell membrane chapter and we talked about the different types of connections? And we had it small because it's not on the AP test. I want to remind you, I actually have that potentially maybe on your exams. So I want to review the 3. Do you remember we talked about in the notes there were tight junctions, which was protein to protein. And very tight, doesn't let anything pass. Something like maybe around your bladder. You don't want things to be leaking out of it into your abdominal cavity. There are gap junctions, which are channel to channel. And there are cell adhesion junctions, which is cytoskeleton to cytoskeleton. So I just want to throw that out there. Maybe take a peek at that or re-listen to this and go over that. Okay. Anything right now where you're walking in here and you have of one of these 4 topics, it's like, what do I need to go over? I will ask you the rest of the clicker questions from chapter 8 that we didn't get to. So I will go visit that in just a moment. But anything like you're super scared about? Okay, so let me just make sure you understand big ticket items. In chapter 5, cell membranes, you want to know the structure of a cell membrane so you could draw it. You could describe the components of it, tell what each of their functions are. You should have that down cold. Okay? You should be able to talk about the different ways to cross a cell membrane, right? Through the phospholipid bilayer, right? But you can't do that if you are what? Large or charged, okay? You could use a channel or a carrier, okay? Or you could use whole membrane like endo and exocytosis. Of those, which are passive, which are active? Through the phospholipid bilayer, that is passive, right? Using a channel, passive. Channel, passive. Using a carrier could be passive if you're going with the gradient, but if you're going against the gradient, it's going to be active. Whole membrane, endo-exocytosis, that's always going to be what? Active. That's always going to be active. All right? Then along those lines, you're going to want to be able to talk about a lab that you did that had to do that. You know, like with diffusion, and there's an essay question that, and how would you handle that? So I'm not going to re-review that. It is on your essay questions, but make sure you feel comfortable with that. Okay. No dying. And then you might want to make sure you understand about how, plants have a cell wall, right, outside of their cell membrane, where we have the extracellular matrix that helps with cell-to-cell junction and recognition. And then what I already told you about, yeah, okay, we should be good there. Chapter 6, energy and enzymes. You want to make sure you're reviewing your first and second laws of thermodynamics. Energy cannot be, what's the first word? Created or destroyed. That's law 1. Number 2, though, every time it changes hands, you're going to lose some as heat. Okay? We talked about the role of passing energy. Who's the first to harvest energy? Auto or hetero? Autotrophs. And then they can pass that on to heterotrophs. Good. When I'm trying to think what else. We talked about ATP, adenosine triphosphate. We talked about exergonic and endergonic reactions. We talked about delta G. Remember all those topics? If you're, cat-like reflexes. If your delta G is negative, think that through for a minute. If your delta G is negative, remember this part, is that going to be an exergonic or an endergonic reaction? Exergonic, because your reactants have less energy. Your products have less energy, right? Okay, because you released energy throughout the process. It is exergonic. Those tend to be spontaneous reactions. They will just happen. What can inhibit them from happening? I'm trying to say it. Energy of activation is that little hurdle that prevents those reactions from occurring. What prevents endergonic reactions from occurring? They require energy because they're endergonic, right? If exergonic could be spontaneous, right? Because you're going from something that has more energy from something that has less. What prevents that from happening? Your hurdle, your energy of activation, yes? What could lower that hurdle? An enzyme. If it's inorganic entirely, then it's just a catalyst, right? Okay? And endergonic reactions are inhibited by the very fact that to be endergonic means you have to input energy into it. The delta G is positive, right? So that's what prevents them from happening spontaneously. Do you see the difference? Any reaction can occur very slowly. But if you want a reaction more likely to occur, you're going to use an enzyme. And if it's endergonic, you're also going to need some ATP. Hence the reason we couple reactions, right? Because an exergonic reaction releases energy, an endergonic reaction could use that energy. So it makes it more likely. So now let's talk about enzymes. Yes?
So an endergonic and exergonic reaction can be coupled together? Or
yes,
okay.
for sure. Coupled together. Okay. All right. So enzymes are made out of what? Protein. So they're organic, right? And what do you call something that is non-protein organic that helps the enzyme do its function? Non-protein organic. A coenzyme. If it's inorganic, it's called a cofactor. Okay? So enzymes are going to be organic, specifically proteins, right? But if it is a non-protein organic, then it is called a coenzyme. You know coenzymes. NAD, NADP, vitamins, those are all coenzymes, right? Bringing it all together. Okay, something like iron or zinc would be a cofactor. Okay, something else you definitely want to know. What are factors that affect enzyme activity? And why do they affect enzyme activity? Temperature, definitely going to affect enzyme activity. Why?
They can denature it.
It could denature
Or
it.
there's a certain rate where at a certain temperature the reaction will go faster because there's more kinetic energy.
Yes, beautiful. He's saying more kinetic energy at a warmer temperature, so that's going to facilitate it, as would any reaction it would facilitate. But they can work best at a specific temperature because it's shape, right? It folds into a certain shape. Change the temperature, you change the shape. Okay? What else can affect enzyme activity? pH. Again, affecting its what? Shape. What else can affect enzyme activity? You did a lab on it. Amount of substrate. Until it reaches a saturated point where all the enzyme's active sites are filled with substrate. It cannot go any faster. What else could affect enzyme activity?
Saturation.
Yeah, point of saturation, we just said that.
Enzyme concentration.
Enzyme concentration can definitely affect it. What else? Big one, big one. Inhibitors. Thank you. What are the two types of inhibitors? Non-competitive and competitive. What does competitive do? Well, yeah, it binds to the active site, blocks the active site, whereas non-competitive binds to the allosteric site, which in turn affects the active site. Okay, Swedish. I like it. All right, so that's chapter 5 and chapter 6. That brings us to chapter 7, photosynthesis. Okay, in photosynthesis, we started off the chapter talking about
chemical and energy, like
energy in general and autotrophs harvesting it.
Yeah, how the energy transfers through photosynthesis.
Yes, that's excellent. And then we focused in on light and wavelengths of light. And what are the things in plants that can absorb wavelengths of light? Pigments. The pigments absorb the wavelengths of light and they can transfer it. Okay, so there's photosystems absorbing all that light and they pass it to the reaction center which starts the light-dependent reactions. Specifically, which one? Photosystem 2. Now, I'm going to tell you, in reality, photosystem 1 goes, photosystem 2 resupplies photosystem 1, and then water resupplies photosystem 2. It does work like that in reality, but it's usually easier for you to understand if we start with the water and work our way through it. That's why I do it like that. Okay? So in photosynthesis, you want to remember there was the light-dependent reactions and the light, what? Independent reactions. Okay? What was another name for the light-independent reaction?
Calvin cycle.
Calvin cycle or the dark reaction. Good. Okay? In the light-independent reaction, there were two. What were the two different ways you could do the light-dependent reaction? Yes, nailing it. Cyclic and non-cyclic. Which one is more efficient? Non-cyclic. Because electrons come in and electrons go out. And because they're going out, who is the final acceptor of those electrons? NADP forming reduced NADP. That is the final electron acceptor. Wow, hi.
Never mind.
Okay, do you,
Come talk to you later.
okay.
Are you leaving early?
I was, but I'll come see you before I go.
Perfect.
Okay,
Okay, thanks.
no problem. Okay, so NADP, the purpose of those electrons is to reduce NADP. In the process of doing the light-dependent reactions, you have some protons. What's the purpose of the protons? The hydrogen ions, protons, right? To form ATP. Yeah, they're not reducing, they're forming ATP, okay? They're going through the ATP synthase complex, yes? So you want to be comfortable with the anatomy of the chloroplast. Remember, you have, okay, on your chloroplast, it's double membrane, yes? Which makes me think about what? The endosymbiotic hypothesis? Is that what you were thinking? Because that's what I was thinking, right? Okay, so this area right here is called the what? Stroma. And these right here are what? Grana. And the grana is made out of what kind of membrane? Thylakoid membrane. Okay. And so the light reactions occur here on the all over them, right? Okay. Sun comes in, right? And you're doing your electron transport chain, and you are concentrating ultimately in the electron transport chain, you are concentrating hydrogen ions to the interior, yes? And when they go back out, you can make some ATP. Good, you make some ATP. Now, also, you pass electrons along your electron transport chains, both photosystem 2 and photosystem 1, and those electrons are going to be given to who? NADP forming reduced NADP. So when you're doing non-cyclic, electrons come in via water and go out via NADP. Who's NADP going to give those electrons to? CO2 is going to get reduced into, yeah, glucose, into carbohydrate. So when you're watering your plants, it's eventually going to use those electrons to build sugar. Because it's going to split the water, right? It's going to take those electrons that we're getting passed along, reduce NADP. It's going to give those to CO2 in order to form sugar. Now, it needs energy to do that because it's an endergonic reaction, right? And it has the sun's energy. And it's going to take that sun's energy, though, and change it into a form it can use chemically, metabolically. So it takes the sun's energy and uses it to concentrate hydrogen ions, which then you allow it to make some what? ATP to do this endergonic reaction. So we say the energy comes from the sun, okay? And you do use that to excite electrons, but you're using that, if you're going to build something in a cell, it's going to cost you ATP if it's endergonic. So you're taking light energy and getting it into chemical energy, into ATP, okay? And then your Calvin cycle, or your dark reaction, you're going to use both your ATP and your reduced NADP, like we said, and making that terrible, your reduced NADP in order to build your sugars. Now your Calvin cycle, how many times are you going to do that? Twice. Why twice? Each time you're going to kick out one PGAL, another name for it, G3P. So we're really putting in 3 CO2s a turn, right? So 3 CO2s come in individually, but they leave connected together. Oh, thank you, sir. And then another 3 come in individually. We hook them all together. If we wanted to, we could take these and build a glucose, right? Okay. Or we could build an amino acid, or we could build starch ultimately with it, right? Any of those things we could build with. Okay. Let's talk about modifications of the theme. We talked about non-cyclic. You could do cyclic, but cyclic is not as efficient, kind of used as backup. And cyclic, you only make what? ATP. Because you don't have electrons coming in and out. Other modifications, we could avoid something we don't like at all. What's it called? Photorespiration. And that's when RUBP hooks up with who?
Oxygen.
Instead of carbon dioxide, and that's not helpful at all. So we can do a partition in space, and we can do a partition in time. Okay? Partition in space are those C4 plants who fix the CO2 and in their mesophyll cells and escort it in via PEP to their bundle sheath cells so that RUBP is never tempted by the fruit of another, right? To partition in time, CAM plants fix CO2 at night. Okay. So that is photosynthesis. Cellular respiration, okay? That's what mighty mitochondria are doing. There are 2 options on that. And respiration. What are they? Aerobic and anaerobic. Which one must we do? What? Aerobic. Why? It gets us more ATP. Okay? You need more ATP. You need to get the most ATP molecules as you can from each glucose molecule. Okay? So if you're going to do anaerobic, you only get how many? 2. And where do they come from?
Where there's 10 and 10, so big and a half.
Glycolysis. Okay? Now, when you do aerobic respiration, you can cash in your reduced NADs and FADs for what? ATP. What I want you to know for the test is each NAD usually cashes out for how much ATP? 3. Maybe new school, they say 2.5, okay? But you are going to know 3 for me, okay? And each FAD usually gives you 2. Maybe 1.5, but it's hard to do the math on that one, okay? So 3 NADs for each strong ATP, you know what I wanted to say? 3 ATPs for each strong NAD, and only 2 for FADs. Only 2 ATP for FADs. Okay, and if you do anaerobic respiration, your first step is going to be what? Glycolysis. And your second step is going to be fermentation. How many choices do we have in fermentation? 2. Either, and why are we even doing fermentation?
To resupply the oxidized
Yeah, to resupply the oxidized NAD. Perfect. Okay? Because if we keep reducing the NAD to make some ATP, we're going to run out of it. The oxidized NAD that we're trying to reduce. So we have to oxidize the NAD. So depending on what kind of organism, you have different strategies for how you oxidize it. You and I, we're going to make what? Yeah, we're going to take our pyruvic acid and reduce it into lactic acid while we're oxidizing our NAD. Right? Redox, one's got to happen with the other. So pyruvic acid gets reduced into lactic acid and you oxidize your NAD. If you are yeast or plants, you're going to take your pyruvic acid and you will reduce it into alcohol and give off a CO2 while you're oxidizing your NAD. If you're going to oxidize one thing, you have to reduce another. Okay? If we have plenty of oxygen though, we won't go down the fermentation pathway. We will move right on into the what organelle will we move into? Mitochondria. What's the next step called? transition prep, and we're going to take our pyruvic acid and oxidize it into 2-acetyl-CoA. And while we're oxidizing it into acetyl-CoA, we'll crank out 2 more. No ATP there. And you reduce some NAD. Yeah. We take pyruvic acid, give off CO2, okay? So we're oxidizing the pyruvic acid, and you reduce some NAD and 2-acetyl-CoA, put 1 away, right? And then we take each of those acetyl-CoA, feed them and hook them up with a 4-carbon to make a 6-carbon for the purpose of just oxidizing it even further, right? And we do the bit. What are we doing when we do the bit?
Take off 1 carbon dioxide.
And you reduce a NAD. How many times in each turn do we do the bit? 2. How many carbons did we bring in? 2. Right? 2 carbons in acetyl-CoA. Do the bit, gets rid of 1. Do the bit, gets rid of the other. And we're back to the C4 again. We make an ATP, reduce an FAD, reduce an NAD, and hook up the other 2 carbon. Do the bit, get 5. There's 1 CO2. Do the bit, get 4. There it is. And then we back to our CO. Now you can account for all 6, because right here's glucose, has how many carbons? 6. We break it in half. We still have 6 carbons, right? Then there's 1, there's 2, right? Then we take this one through, do the bit, take this one through, do the bit, sorry. Okay, so that's all 6 carbons right there. Do you follow it through? Okay? Yes?
So when you're doing the bit, what actually is that process? I know.
You're reducing the NAD and pulling off the CO2. Yeah. It's the same thing as transition, but you're not putting a coenzyme on it. Okay? Then following the Krebs cycle, we do the, what do we call it? Electron transport chain, ETC. We gather all our reduced NADs and FADs. Where did we make all those NADs and FADs? We made 2 NADs in glycolysis. We made 2 NADs when we did transition prep. Each turn, we made how many NADs? Do the bit, and then we reduce an NAD. We made 3. We do the turn twice. That gives us 6 more, right? So we have 6 NADs from the Krebs cycle, 2 NADs from transition prep. We have 2 NADs from glycolysis. Okay, so that's 6, 7, 8, 9, 10. I count 10 NADs, right? If those gave us 3 ATP each, we would have how many? 30, right there. You see that? When we reductase, cytochrome, oxidase. But the NADs in glycolysis, they sometimes get you 3, sometimes they give you 2, because they pass it on to another player. Who is the other player we haven't discussed yet? FAD. How many FADs do we make a turn? 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD. We get 1 a turn, and we do it twice. So 2 times 2 is 4 ATP. Plus, don't forget about your substrate level phosphorylation and glycolysis. You get 2 from there, too. Okay, I'm not having you account for all that. I just want you to see how you're in the high 20s and the 30s in order to get your ATP from aerobic respiration. Okay, questions? Yes?
FAD used to reduce during the bit, only NAD, right?
FAD is happening after you've already done the bit. Yeah. Okay, questions or concerns about that? Okay, let's do a few quicker questions then. Three, 1, 4. What do you think the answer is? Oxygen doesn't contribute any electrons, so I hope you're not picking that. Glycolysis to the electron transport chain, it contributes 2 NAD reduced, and oftentimes it gives them to the FADs, right? So that's not going to get you very much ATP. Citric acid cycle, what does it give you? 1, 2, 3 NADs times 2 turns of it gives you 6 NADs, and it gives you 2 FAD reduced. I would say that gives you the most, because the prep reaction only gives you 2 NADs reduced. Do you see that? Wow, some of you saw that. Okay, so let's just talk through this. Substrate level phosphorylation takes place where? When do we do where you aren't, when you're making ATP but you're not nine? Where do you do that? Glycolysis. Where else do you do it? Citric acid cycle. So where is that? A, yes. Okay? That's when it's a direct handoff. If you're not doing substrate level phosphorylation in the electron, you're doing oxidative phosphorylation. No. Who catches electron on the bottom? Oxygen forming what? Water. Okay? What's your answer here? Water. Yeah, it becomes water. Good. We already did this just now, right? 6 from the citric acid cycle, 2 from transition prep, and 2 from glycolysis. 6, 7, 8, 9, 10. 10 NADs, right? 2 from glycolysis, 2 from transition prep, 6 from the 2 turns of the Krebs cycle. How many are usually produced? 3. Really 2.5, but okay, and then we already did that part. And that would be the end. Good job, you guys. Study up, watch all your videos, review, write your essays, be smart. Remember to review your cell junctions.